Good evening, everyone. I'd like to welcome you to the 1,251st meeting of the Entomological Society of Washington. I am the interim MC tonight. Our president is on vacation. Past president is ill. President-elect wants to see how this all works. So I've decided to step in and do it as past president and your current membership secretary. Last officer standing. You're an officer too, you know. Well, that's right. Yeah. So, okay. So I'm gonna read. Okay. Okay. I would like to ask right now, recording secretary Gary Hevel to read the minutes of the last regular meeting, please.
The 1250th regular meeting of the Entomological Society of Washington was called to order at 7 p.m. on February 6, 2025 in room WG33 of the National Museum of Natural History. President Andrew Jensen greeted members, visitors, and virtual attendees and was appearing himself in virtual format. 9 members, 9 visitors, and 25 virtual participants were in attendance. Recording Secretary Gary Hevel read the minutes of the January meetings, and these were approved by the audience. New member David Frischback, a student at Churchill High School, introduced himself, noting that his specific entomological interest is weevils. He was accompanied by his mother, Patricia Frischback. Manon Davis held up a copy of the October 2023 issue of the Proceedings of the ESW, noting that it was the last issue present in the entomology library at the museum. As a former ESW membership secretary, Manon noted that hard copies of the proceedings serve as advertisements globally and that all libraries should have currently available printed copies. Curator Alyssa Seaman commented that she will provide recent issues to the library. Membership and Communications Officer Matt Buffington noted that he and Webmaster Lourdes Tomorrow, during their recent attendance of the International Congress of Entomology in Japan, found many items of interest, including insect-shaped candies, a book of the Vespidi of Japan, and a volume entitled The Japanese Book of Beetles. President Jansen suggested positioning these items for view by a virtual audience, and Matt followed through on this. Matt also mentioned the copies of the revisionary publication on the Sericity by David Smith are available for anyone interested in that subject. Webmaster Lourdes Chamorro directed the attention of the audience to a table where copies of various entomological books and articles were placed. These items were available to anyone with interest in them. In exchange, monetary contributions to the society were welcome. Such volumes and articles will be offered at future meetings, and it is hoped that this will stimulate members and visitors to attend meetings in person. Program Chair Alan Norbaum introduced Evan Economo as the speaker of the evening, whose presentation was titled Mandibles 3D, Unraveling the Evolution of Hooks, Drills, Straps, and Other Diverse Tools. Mandible diversity is vast within ants, with the basic triangular design of the head. Trapjaw mandibles have power amplification, and the action can be compared to the well-known mantis shrimps. Timing comparisons between trapjaw ants and mantis shrimps have concluded that the ants are fast. The genus Strumigenus is known to feed upon springtails, and the first group of ants studied by E.O. Wilson. Springtails are grasped, then stung by these ants. 1/3 of individual ants have short mandibles. Several evolutionary pathways have taken place, but results are always within 3 types. Measured speed indicates that trap jaw ants close their jaws 5,000 times faster than the blink of an eye. The ant genus Melissotarsus lives in live wood, as opposed to all other ants that live in dead wood. Individuals of the genus have large, fat legs and can move with middle legs moving against the top of tunnels. Synchrotrons are being used to provide information on comparative body systems. An ant scan project in Japan using a 30-second process scanned 2,200 specimens of 850 species in 1 week. 3D models allow large-sized images with horse-sized images, and insights are gained from this activity. Other study methods are being developed, and there's much to be learned in the future. The meeting was adjourned at 8:35 p.m.
Are there any additions or corrections to the minutes as read? Hearing none, can I have a motion to have them accepted as read?
I vote to
Okay,
accept.
in a second. So approved. Thank you, Recording Secretary Hubble. Okay, next up we have reports of officers and committees. Officer Norbaum, do you have anything to report?
Our next speaker is going to be Jeffrey Sotelo.
Okay. Yeah.
April 3rd meeting.
Something about ants, probably.
But yes,
Yes. President-elect Samois, anything to report to the group?
We have a speaker for the annual banquet. It's gonna be Ted Schultz
And where are we thinking of holding the banquet?
at Best Buys and Poets in Chinatown. So not a potluck this year, sorry.
But easy to access, metro accessible,
Yes,
and booze.
yes, and we are all tired, so let's have some activity for us.
Cool. Okay. Recording Secretary Hubble, do you have anything to report?
Nice.
Okay. Anyone? Alyssa Siemens, do you have anything? I just saw something that popped up and disappeared. What did she say?
I just said it would be nice and museum accessible as well.
Can you repeat that?
I just said it would be nice and museum accessible as well.
Yep. Okay. Okay. I will give anyone else online with a report. Any other? Nada. Okay. As your membership secretary, with Dr. Tomorrow's help, I successfully navigated the Entomological Washington website, which reveals membership information. I have used this to compile the following list of new members, which have to be read in at a regular meeting before they're approved at the next meeting. So far, we have added, since I had no one to read names at the last meeting, this is the last 2 months. So this includes 5 people. Zachary Gribenow, Addison Copen. Some of these are like back, okay. Oh boy, this one I'm really having trouble with. Berku Jimeshk. It has like certain characters. Maybe someone else could read it at some point. Mariella Oviedo and Brad Barnes. So these are 5 new members of society, not to be confused with just renewals. So I'm really excited to have new people in. And I think that's it for that. Okay. If there's a new member in the audience or a visitor in the audience, please introduce yourselves.
Hello, I'm Cody. I use they them pronouns and am excited to be here. Longtime insect fan.
Awesome. We're excited to have you.
Great to have you.
In the back.
It's Catherine Turk. I'm a postdoc in paleobiology here. I'm a speaker from college. We're old friends.
Excellent. Cool.
I'm also listening on Baron Linnick. I'm a postdoc in Cascade. Heard about the event from?
Awesome. Fantastic. Cool.
Yeah, that'd be great.
Is there anyone out in Zoom land that would like to introduce themselves? There's Robin Marquez. He's a regular visitor, former intern of mine, so. Anyone else want to say hi? Turn on your microphone and say hello. Everyone's just super excited to get.
Shy Zoom.
They're shy Zoomers. They're excited to get to business. Okay. So, is there any unfinished business from the last regular meeting? Or any new business someone would like to introduce? We're following Robert's rules, by the way. If you're not familiar, this is how parliamentary things work.
Abby said there was another step on the corporation.
Yeah, we had to give the officer addresses, so
Reaching closer.
it seems like we're at that plateau that we're never quite there, but we've been forming or formally being recognized as a 501(c) now for like 10 years, so we're finally getting there. Presentation of notes and exhibition of specimens. Does anyone have anything to share? This bounty of giveaway books is from our own Gary Hebbel. These I have brought for him down here, free for you guys to take. So please have a look. And we still have more Cyricid woodwafts of North America keys. So I've got about 80 more of them. So I just keep bringing them. I brought 3 more today.
I have a book.
Hey, okay, great.
This was from Don's collection in the office. Biology of the Leaf Miners by Eric Martin Herring. It covers Lepidoptera, Diptera, Hymenoptera, and Coleoptera,
Cool.
and has a few good illustrations in it.
It's a good book. I have a copy.
Yeah.
Oh,
You want to pass it around, or
Yeah.
is it too precious?
no, I'll.
Just be careful with it. Don't get pizza sauce on it.
Around.
Cool.
I have pizza on my.
Thank you, Manal, for bringing that up.
Okay.
Any other exhibitions? Okay, I think I'm ready to turn it over to Program Chair Norbon.
Here we have 20 people on Zoom.
Yes, and 22.
Well, that counts.
Oh.
This one at least.
Yeah.
Can't count ourselves twice.
No.
Okay, I have the pleasure to introduce tonight's speaker. We're continuing our University of Maryland run here. Morgan Thompson is a postdoctoral research associate in the Department of Entomology at the University of Maryland, working with Dr. Karen Burghardt, former and collaborators at the Smithsonian Environmental Research Center. She earned a BS in biology from the College of William and Mary, go Tribe, and completed an MS in entomology with Dr. William Lamp at the University of Maryland. She earned a PhD in entomology under Dr. Angel Helms at Texas A&M. Her research program integrates chemical, molecular, and community ecology to investigate plant-insect interactions. Specifically, she studies the chemical mechanisms and ecological consequences of plant defense against above ground or below ground insect herbivory, with the goal to generate new understanding of plant anti-herbivore defense in order to provide sustainable pest management strategies for natural and agricultural ecosystems.
All right, great. Thank you so much. I think everyone here can see the PowerPoint, and then hopefully folks on Zoom
can also see the PowerPoint if you can't. Chime in. Okay, we're good. Great. All right. Well, thanks so much for the introduction, and thank you all for the invitation to be here today. I'm so excited to talk with you all a little bit about my research. And so just to give you all an outline for my talk today, first I'm going to go over a little background about myself, which Alan just covered, but I'm going to go over it again. And then I want to tell you 3 different stories today from my research program. And then finally, I'm going to wrap up with some conclusions and then future directions for some of my work. Okay, so to get started, a little background about me, about the research topics that I'm interested in. So I'm originally from New Jersey, born and raised, and I completed my undergraduate degree at the College of William and Mary. And yes, indeed, go Tribe. And so I got my degree in biology. And it was there that I was introduced to a lot of really exciting topics. So ecology, evolutionary biology. And I was fortunate enough to land in a lab that was studying butterfly population dynamics. And so that's what I learned about entomology. And my research advisor at the time, he told me, he said, you know, Morgan, if you're really interested in entomology and research, there are entire departments at bigger universities devoted to entomology. And that just blew my mind at the time, coming from a really small school. And so I decided to go on and pursue my master's degree in entomology at the University of Maryland. So it was there that I worked with Bill Lamb. I got introduced to agricultural entomology. I was working on alfalfa leafhopper interactions. And it was also at the University of Maryland at that time that I got introduced to teaching. And I really loved teaching, research. And so I knew that the next step then was to go on for my PhD in entomology at Texas A&M University, where I worked with Angel Helms. And it was here that I was working with gourd plants, which is what I'll be talking with you all about today. So these are the family Cucurbitaceae. And I had a lot of fun experiences in Texas. I really enjoyed my time in College Station, do some really awesome hiking. I learned a lot about football. When you move to Texas, that's a thing that you do generally down there. I also got introduced to Buc-ee's. Are people familiar with Buc-ee's? This is like the craziest place ever. It's a convenience store slash kind of like Walmart thing down in Texas where they have this lovely mascot, Buc-ee. And also I had a lot of really good barbecue. So if anyone wants barbecue recommendations in the Houston, Austin area, I can help you out. So yeah, so that's a little bit about me. So now I want to switch gears and talk a little bit about my research. So we know that when this caterpillar begins feeding on a plant, plants are not defenseless in these interactions. And actually, they have these really sophisticated mechanisms for protecting themselves against insect herbivory. And we broadly classify these different plant defense strategies into 2 separate categories, the first being resistance, and then the second being tolerance. And so I want to talk a little bit more about plant resistance and tolerance against insect herbivory. So when we think about plant resistance, there are a number of different mechanisms that plants can use to protect themselves against insect herbivores. The first being physical defenses. So these could be things like spines on the plant that will provide oftentimes a direct defense against herbivory. So that could be either repelling foraging herbivores that check out a plant that's really spiky and spiny and say, no, thank you, I don't want to feed on that host plant. Or they can deter herbivores that are already directly feeding on that plant, often by impaling an herbivore. But plants also have lots of different chemical mechanisms that they use to protect themselves against insect herbivory. And that'll be the focus of my talk today. And so these chemicals are often produced endogenously within the tissues of the plant, but they can also be emitted outside of the plant. And we'll be talking a lot more about those volatile chemicals that plants emit throughout the talk. And these can provide a direct defense for plants, but they can also play a role in what we call indirect defense of plants. And that's when plants recruit predatory or parasitic insects to come kill herbivores feeding on that host plant. So this is generally how we think about plant resistance against insect herbivory. But then kind of on the opposite end of the spectrum, we also have tolerance. So this is when essentially plants regrow to replace the tissue that's been damaged by herbivores. But Oftentimes folks will also categorize shifting of nutrients within a plant, so reallocating nutrients away from herbivores as a form of tolerance against insect herbivory. Okay, and so we know that these different plant defense strategies are happening at different scales within a host plant. So when this caterpillar begins feeding on this leaf tissue, there are molecules in the saliva of the caterpillar that we term elicitors, that plants are actually really good at detecting and figuring out exactly which type of insect herbivore is feeding on them. And then following plant detection of specific insect herbivores, this activates defensive signaling cascades within the plant that ultimately lead to the increase in these toxic chemical compounds within plant tissues, as well as these herbivore-induced plant volatiles, or HIPVs. So these defensive chemicals that plants are emitting outside into the atmosphere. And again, we'll talk more about HIPVs as we go along through the talk. So this is what we generally think about in terms of what's happening on the local scale within a plant and how it's activating defenses against insect herbivory. But plants are also responding systemically to insect herbivore damage. So we know that when this above ground herbivore begins feeding on a plant, this can systemically change what's happening in the below ground root tissues of the plant. So this can alter the metabolism or the physiology of the root tissues. And conversely, we know that when below ground herbivores begin feeding on root tissue of plants, this can also alter plants systemically and change above ground tissues. Okay, so with all of this background information in mind, this leads us into the first story that I want to tell you about from my research program, and that's what I'm calling an undercover operation today. And so when I began my dissertation research, one thing that I found really fascinating is that there are a handful of insect herbivore species that feed both above ground and below ground across different life stages. So oftentimes the larvae are feeding below ground, such as in this example with these weevils feeding on clover. And the adults are then emerging and feeding above ground on the foliar tissue of plants. And so I find this really interesting because it sets up these plant-mediated interactions between different life stages of the same species of insect herbivore. And so the handful of studies that have previously investigated this in the literature have actually found instances of competition across these different life stages. So in the rice and raspberry system, when one insect herbivore begins feeding either above or below ground, this has a negative effect on the growth or the performance of a conspecific feeding on that other plant compartment. But interestingly, we also see examples in the literature of the exact opposite plant-mediated interaction between these conspecifics. And those are examples of facilitation in the Chinese tallow and the milkweed system. So essentially, when above ground herbivores begin feeding on these plants, they enhance the performance or the growth of the below ground larvae that are feeding on the root tissue. And so what I realized going into my dissertation work is that I had a really nice study system to investigate some of these plant-mediated interactions between above ground and below ground insect herbivores. And that's with the cucurbit system. So specifically for this project, I was focusing on zucchini squash plants, which are one species of plant in this gourd plant family. And I was looking at below ground striped cucumber beetle larvae. So these are specialist insect herbivores, which means they feed exclusively on plants in the cucurbitaceae. And these below ground root feeding herbivores are particularly problematic in agricultural systems. When the plants are really young, they can cause significant damage. So these below ground root feeding larvae feed on the root tissues of plants, pupate in the soil, and then emerge as adult beetles and feed on the above ground foliar tissue of the plants. And these beetles can also be really problematic in agricultural systems because they can transmit a plant pathogen. And then I was also interested in looking at squash bugs, which are another specialist insect herbivore of cucurbit plants. But these are piercing sucking herbivores, and they feed as both juveniles and adults on that above ground foliar tissue of plants. And they can also transmit a plant pathogen. So they're also really problematic agricultural insect pests. But what we didn't know going into this research is really how plants navigate being attacked both below ground and above ground by these different insect herbivores. And so that was the question that I wanted to ask in this study is, can below ground herbivory alter systemic plant defense? And the overall goal being here to determine how below ground herbivory alters above ground systemic plant defense and then interactions with these above ground insect herbivores. And based on some previous research in the lab, I predicted that these below ground herbivores would systemically enhance plant defense and thereby deter these above ground competitors. So to test all of this, I first hypothesized that above ground herbivores would avoid below ground larvae damaged plants. So I set up above ground herbivore plant preference assays in the lab where I introduced 2 different plants into a mesh cage. I had my control non-damaged plants relative to my below ground larvae damaged plants. And for my adult beetles, I would then introduce 6 starved female beetles into each cage. And then after 48 hours, I would come back and record where I found the beetles, as well as the amount of feeding damage that they inflicted on these plants. And then for my squash bugs, I again gave them the same choice between these control and larvae damaged plants, but this time I introduced 2 mating pairs into each of the cages. I allowed the bugs to feed and oviposit for 2 weeks, at which point I came back and recorded where I found the bugs and counted the number of eggs that they laid on either plant. Okay, so what did we find? So first, I'm showing you on the x-axis here the number of adult beetles, and in those white bars, the beetles that were found on the control plants relative to in the yellow bars, the beetles recovered on the larvae damaged plants. And what you can see is that I found many more beetles on those control plants relative to the larvae damaged plants. And very similarly, when we looked at feeding damage, although non-significant, I found a trend towards more feeding damage on the control plants. So it seemed that these conspecific adult beetles were really avoiding those larvae damaged plants. And then very similarly for squash bugs, here I'm showing the number of squash bugs found settling either on control plants or larvae damaged plants. And we found many more squash bugs on those control plants relative to the larvae damaged plants. And very similarly, the squash bugs preferred to lay their eggs on those control plants relative to larvae damaged plants. So overall, it seemed that conspecific and heterospecific above ground insect herbivores were really avoiding those below ground larvae damaged plants. So that was very interesting. But then the question, at least for me, was, well, why? Why are they avoiding these plants? What's the mechanism of this host plant selection behavior? And so I then went on to hypothesize that perhaps below ground larvae feeding damage was altering the above ground plant volatile emissions. So these chemicals that the plants are emitting from their leaves, they can be really important for herbivores that are selecting different host plants, because they can signal to these herbivores the quality of the host plant oftentimes. And so I hypothesized that perhaps the larvae were changing the volatiles that the leaves were emitting. So to test this hypothesis, I set up above ground plant volatile collections where I had 2 different groups of plants. I had my below ground larvae damaged plants relative to my non-damaged control plants. I allowed those larvae to feed for 48 hours, and then I collected the above ground volatiles that were emitted from the leaves of both of these groups of plants. And to do that, I used dynamic headspace sampling, which you can see here. Okay, so what did we find? Well, we found that one key volatile compound was really changing, and that is E-beta-ocimine. And so here on the y-axis, I'm showing the abundance of E-beta-ocimine, and on the x-axis, I'm showing the amount of E-beta-ocimine emitted by the control plants relative to the larvae damaged plants. And what you can see here is that when we add those below ground root feeding larvae, we get this big increase in the amount of E-beta-ocimine that the plants are emitting from their leaves. And so this was a really exciting finding. And I thought, oh, yes, this is the chemical signal that these above ground herbivores are honing in on, and that's how they're selecting their host plants. But spoiler alert, when I went to test that and looked at their behavioral responses to ocimine alone, that was not the case. So it seemed that actually this chemical cue, we didn't find strong evidence for it really driving the foraging behavior of these herbivores. So then the question became, well, what is the mechanism then of this host plant selection behavior? So I then went on to hypothesize that perhaps below ground larval herbivory was modifying the above ground nutrients in the plants. And so non-structural carbohydrates, so essentially the sugars in the plants, as well as the protein content of plants, can be really important for herbivores when they're selecting which plant they want to feed on. So that's where I decided to look next. And so to test this question, I set up 2 different groups of plants. I had my larvae damaged plants relative to my non-damaged control plants. And I collected leaves from all of these plants and then quantified the above ground protein and carbohydrate content using colorimetric assays. Okay, so what did we find? So first I'm showing the results for carbohydrates. So this is the carbohydrate content of my control leaves in the white bars relative to my larvae damaged plants in those yellow bars. And so what you can see here is that although not significant, we found a trend towards a reduction in the carbohydrate content of that leaf tissue when we added those below ground root feeding larvae. So that was really interesting. But then even more interesting is that we found the opposite pattern for protein. So we actually found this increase in the protein content of the leaf tissue when we added the below ground root feeding larvae. And then when we looked at the ratio of protein to carbohydrate content, which can be, again, important for herbivores when they select their host plants. It might not be about the absolute quantity of one nutrient versus the other, but rather the ratio. We again see the shift when we add the below ground larvae. And so when we put all of this together, and and if you want to read about it, you can check out my publication, where there's a little bit more information, a few more tidbits there. But when we put all of this information together, what we found is that when we add these below ground root feeding larvae, these above ground herbivores are avoiding those plants, so both the adult beetles as well as squash bugs. We also found that these larvae are causing the plants to produce more E-beta-ocimene above ground, but we couldn't link the foraging behavior of those above ground herbivores to E-beta-ocimene. So perhaps it's their behaviors are driven more so by these gustatory cues, so these changes in the nutrient content of the leaves. I think we need to continue on with some of this work to really hone in on if that's the exact mechanism. But I think this is some strong evidence pointing towards these nutrients. And so that kind of summarizes my first story. But it did leave this lingering question in my mind, all of this work. And that was really about E-beta-ocimene. And so I was wondering, well, what is E-beta-ocimene doing in this system? If it's not repelling these above ground herbivores, what could it be up to? And that leads us into my second story tonight, which I'm calling eavesdropping plants. And so as I mentioned earlier, there are a number of different functions of these herbivore-induced plant volatiles, or HIPVs, and that's in direct defense and indirect defense of plants. But there's a third function of these HIPVs that I didn't tell you about, and that's in plant communication. So we know that when this plant is being fed on by a caterpillar, it's emitting this blend of HIPVs that a neighboring plant is actually really good at detecting and using to enhance its defenses should that caterpillar start wandering over and feeding on that neighboring plant. And so this leaf-to-leaf communication between plants has been documented for many different plant species attacked by many different insect herbivores. But something that I began wondering about is, well, what about these systemic HIPV emissions? So we have this system with the zucchini squash where we know that when we introduce these below ground root feeding larvae, they're changing these systemic HIPVs, or the chemicals that are emitted by the leaves of the plant. And so it was unknown at the time if these systemic HIPVs could play a role in plant-plant communication. And so I convinced a group of undergraduate students that this was a cool thing to investigate. And so through the Texas A&M Aggie Research Program, I mentored a group of 4 undergraduate students in a team research project to ask this question. And what we wondered was, can below ground herbivory induce plant communication? And our overall goal here was to evaluate how systemic HIPVs from below ground larvae damaged plants modify above ground defenses of neighboring plants. And based on that prior work, what we hypothesized here is that perhaps neighboring plants are responding to these systemic HIPVs. So if the neighboring plants get a whiff of those chemical cues, they're better defended against both these above ground beetles and squash bugs. Okay, so how did we test this question? So we set up plant communication experiments. And for these experiments, we had 2 different bins of plants. The first being control bins, where we had two different types of plants. We had emitter plants, which were our plants doing the talking in this experiment. And then we had 2 receiver plants, which were the plants doing the listening in this plant communication experiment. And so we compared these bins to our HIPV bins. So here are 2 emitter plants or larvae damaged plants. And so we allowed all of these plants to talk to one another for 24 hours. Then we came back and we removed all of the emitters from the bins. And then we challenged those neighboring receiver plants with adult striped cucumber beetle herbivory. So we were interested in looking at the response of the receiver plants following these different exposures. Okay, so what did we find? So here on the y-axis, I'm showing the amount of feeding damage from those adult striped cucumber beetles. And on the x-axis, I'm showing my control receiver plants relative to my receiver plants that were previously exposed to those systemic HIPVs, or the larvae damaged plants. And what you can see here is that we found really no difference in the amount of feeding damage by those adult beetles, which indicated to us that prior exposure to those systemic HIPVs really did not change the defenses of those neighboring plants. And so this was a bit disappointing. It did not match our prediction, but we were still wondering, well, what about neighboring plant defenses against squash bugs? Will we see a similar response if we set up those same plant communication experiments, but this time we challenge those neighboring receiver plants with squash bugs instead of these adult beetles? And so that's what we did, and we found something a little bit different. So here on the y-axis, I'm showing the mass gain of the squash bugs, and on the x-axis, I'm showing those control receiver plants relative to those receiver plants that were previously exposed to those systemic HIPVs. And what you can see here is that the squash bugs feeding on those HIPV-exposed receiver plants gained less weight or mass than the bugs feeding on the control plants, which indicated to us that those plants that were previously exposed to those systemic HIPVs were more resistant to squash bug herbivory. And so this was a really exciting finding, but we didn't quite stop there. And then we were wondering again about osamine. So what if we just expose these plants to osamine alone, to just this one chemical? Can we see that same effect in our plants? So if the plants get a whiff of osamine, are they better defended against squash bugs? And so to test this experiment, or to test this hypothesis, we set up osamine exposure experiments where we again were putting plants in bins, but this time we had our control bins relative to our osamine bins. So here you can see the students setting up these experiments, and we had these osamine dispensers that we were using to expose those plants to this specific chemical. And so after 24 hours, we came back and removed all of the dispensers from the bins, and then we challenged those neighboring receiver plants with squash bug herbivory again. And here's what we found. So here on the y-axis, I'm showing the mass gain of the squash bugs, and then on the x-axis, I'm showing those control receiver plants relative to the receiver plants that were previously exposed to osamine. And what you can see here is that we again found this reduction in the mass gain of the squash bugs on those plants exposed to osamine, which told us that that prior exposure to osamine was enhancing the resistance of these zucchini squash plants against squash bug herbivory. And so when we put all of this together, when we expose neighboring receiver plants to these larvae damaged plants, we find that this exposure to the systemic HIPVs did not alter neighboring plant resistance against adult beetle herbivory, but it did heighten neighboring plant resistance against squash bug herbivory. And then we further went on to demonstrate that osamine seems to be serving as a key chemical compound eliciting that neighboring plant resistance against squash bugs. And so if you'd like to read more about this story, you can check out this other publication with myself and the students. And yeah, so that wraps up story number 2 and takes us into my final story for the evening, which I'm calling phytochemical diversity. And so here we're going to be talking about all the different types of chemicals that plants are producing. And we're going to switch gears a little bit from thinking about systemic plant defense responses to going back to those local plant defense responses. All right, so we know that when this caterpillar begins feeding on a plant, it's changing plant volatile emissions, as we've talked about a little bit this evening. And there are a couple characteristic changes that we typically see in the volatile blends that plants are emitting. And the first is an increase in the quantity of volatiles following this insect feeding damage. So the plants begin to produce more volatiles after herbivory. But we also see this change in the types of volatiles that plants are emitting. So oftentimes they'll increase the numbers of different chemical compounds that they emit after insect herbivory. So we get these quantitative and qualitative changes in the HIPP blends. And so something that I found really interesting going into my PhD research is that plants do this both above ground and below ground. And this has been really well studied in the corn system. So we know that when we add a caterpillar to corn leaves, these plants are emitting this characteristic blend of volatiles that's attractive to parasitoids. But interestingly, corn plants also do this below ground. So when rootworms begin feeding on the roots of corn plants, they're emitting chemicals into the rhizosphere that are actually attractive to enemies of those rootworms. And so this has been very well studied in the corn system. But what I realized going into my dissertation research is that beyond corn and a handful of other study systems, we don't really know much about the patterns of HIPV emissions above ground and below ground across different plant species. And so that's the question that I wanted to tackle with this project and ask, how do HIPVs compare across above ground and below ground systems? And the overall goal here was to characterize foliar root HIPVs of cucurbit plant species to compare volatile diversity across these different systems. And my two specific objectives here were determining first those quantitative changes in the volatile blends following insect herbivory, and then also to identify qualitative changes in volatiles in response to herbivory. Okay, so to test this, I was working with cucurbits still. Again, plants in the gourd family, and I had 6 different plant species for this particular project. I was working with 4 agricultural crop species, so those were pumpkins, cucumbers, zucchini, squash, and watermelons, as well as 2 wild gourd species for this project. And then for my above ground insect herbivore, I focused on squash bugs for this particular study, as we've discussed in these previous projects. And then for my below ground cucurbit insect herbivore, I was working again with strict beetle larvae. And again, interested in the local plant defense responses, in this case against these herbivores. Okay, and then here I'm showing a phylogeny of all 6 of these different plant species to show their relatedness to one another. And so you can see here that we had a few different genera represented in this study, and some species that were much more closely related to one another, such as the zucchini squash and Texas gourd, which is a wild gourd relative of zucchinis. They're much more closely related to one another than they are to cucumbers and watermelons, for example. Okay, so how did we do this? I set up some leaf volatile collections. So again, I was using dynamic headspace sampling here, and I had 2 different groups of plants. I had my control non-damaged plants relative to my squash bug damaged plants. So I allowed those bugs to feed for 48 hours, and then I collected the volatiles from both of these groups of plants. And I repeated these experiments for all 6 different plant species. Okay. And then very similarly, I set up root volatile collections to look at plant responses to that below ground herbivory. So again, here I had 2 different groups of plants. I had my control plants relative to my below ground larvae damaged plants. And I set up dynamic headspace sampling where I collected the volatiles from both groups of plants after 24 hours of larval herbivory. And I again repeated these experiments for all 6 of my plant species of interest. Okay. So what did we find? So thinking about that first objective, looking at these quantitative changes in volatile emissions after herbivory. So here I want to focus first on the foliar volatile emissions following squash bug herbivory. So what I'm showing on the x-axis are all 6 of my plant species of interest. And then on the y-axis, I'm showing what I'm calling total volatile induction. So this is a ratio that I calculated from each herbivore damaged plant relative to the average baseline non-damaged control plant for each plant species. So essentially a value of 1 for this ratio would tell us that our herbivore damaged plant is emitting the same quantity of volatiles as the average non-damaged control plant for that species. And then any value above 1 tells us that an herbivore damaged plant is increasing the quantity of volatiles that that particular plant is emitting. So what you can see here is, as we predicted, we see this increase in the volatile emissions when we add those above ground herbivores across all 6 plant species. And there's some variability across the species, but in general, when we add an herbivore, we get an increase in the chemicals that the plants are emitting. So that was really interesting. And then when we looked at root volatiles, we found a very similar pattern. When we add that below ground root feeding herbivore, we get this increase in the quantity of volatiles that the plants are emitting from their roots. And we see some variability across species. But the thing that I want to hone in on here is the difference in the y-axis for these two graphs. And what you can see here, and what this is telling us, is that the induction is much stronger above ground than it is below ground when we add those herbivores. So this was a really interesting pattern that hadn't been documented in the literature before. And so that was the story for these quantitative changes in the volatile emissions. But then what about these qualitative changes in the volatile blend following herbivory? And so here's what we found in that regard. So first, I just want to focus in on the zucchini plants. And here what I'm showing are my control leaves relative to my squash bug damaged leaves, and then my control roots relative to my larvae damaged roots. And on the x-axis here, I'm showing volatile richness, which is a measure of the numbers of different types of chemicals that the plants are producing. And the pattern that I want you to really focus in on here is the change when we go from control leaves to our bug damaged leaves. We see this strong increase in the numbers of chemicals that the plants are producing. But then in contrast, we really don't see much of a change below ground when we add those herbivores. And then what was really interesting is that when we looked across all of our plant species, we found a really similar pattern. So when we add those above ground herbivores, we get this strong increase in the numbers of chemicals that the plants are emitting, but we don't see that same pattern below ground. And so again, this was something that hadn't quite been documented in the literature before. So we were very excited about this. And so when we summarize all of these findings, what we found is that these quantitative changes in plant volatile emissions were stronger above ground relative to below ground. And then we also found that herbivory was altering volatile richness to a greater extent above ground than below ground. And so I think these findings are really exciting in terms of thinking about what these HIPs could be up to in these different systems. So it's possible that things like indirect defense, for example, where the plants are calling in the natural enemies to help them defeat these herbivores, that could be more important above ground than below ground. But these are questions that we would have to test in the future. So yeah, and if you'd like to read more about this work, you can check out this recent publication where we get into some other patterns and findings. Okay, and then finally, so this final story that I'm telling you today, it did leave me with this lingering question though. So these roots are not strong inducers of volatiles. We saw that with that last study, but then how are they protecting themselves against insect herbivory? If they're not using these HIPVs, what could they be doing? I won't have time to really get into it today, but the exciting part is that it seems like they're relying more on tolerance below ground. So these roots are much better at tolerating herbivory than the leaves. And yeah, and I just got revisions back on this manuscript, so this will be coming soon. But yeah, this just to give a quick summary of what we found here. It seems that the roots are more tolerant, so they're regrowing after herbivory. Okay, so wrapping up with some conclusions. First, we find that these local plant defense strategies against above ground and below ground insect herbivores are distinct. So it seems that above ground plants are relying more on resistance through these HIPVs, perhaps, and then below ground, the roots seem to be tolerating herbivory, so regrowing after herbivore damage. Oh yeah, and then I noted that in the PowerPoint slide. So leaves are resisting herbivory, roots are tolerating herbivory. And then in terms of these systemic plant defense responses, we documented that these are mediating these different interactions across both above ground and below ground ecological communities. Okay, but I think there's some interesting outstanding questions from some of this work, particularly on the systemic plant defense side. So as I mentioned earlier with those first two stories, what we found is that these below ground root feeding larvae are perhaps maybe really good at manipulating their host plant. And I want to tell you what I mean by that. So we know that they're really good at getting these above ground herbivores away from the host plant. So these competitors, perhaps, they're repelling them. And we also know that these plants are then emitting more E-beta ocimine above ground, and that this is playing a role in strengthening plant defenses against squash bugs, but not adult beetles. And so perhaps these larvae are really good at manipulating even neighboring plants to make those plants better for their future adult selves, but worse for their competitors, these squash bugs. So just some interesting things to think about what could possibly be going on in these ecosystems. And so then finally, I want to wrap up very briefly with some future direction. So what I'm up to now. So I'm back at the University of Maryland. I'm super excited about that as a postdoc. And I'll be working in collaboration with folks at the Smithsonian Environmental Research Center on the biodiversity tree experiment, which is a really exciting experiment where there have been 15 different temperate tree species growing in different plots, in single species plots, so essentially monoculture plots where you just have one species of tree surrounded by the same exact species, versus mixed species plots where you have a tree species surrounded by a bunch of different tree species. And folks have for over a decade now been tracking trees growing in these different environments. And so there are a number of really exciting data sets that have been generated from this experiment. And so with all of these different tree species, folks have been looking at the chemicals that the leaves are producing, which I, as a chemical ecologist, find really exciting, is thinking about how plant chemistry is changing in these different environments. But also, folks have been looking at the microbial communities in the leaves of the plants and how, again, they're shifting in these different environments over time, as well as the root microbial communities associating with the roots of those trees. And also tracking insect herbivore communities feeding on the leaves of these trees growing in different environments over time. So I'm very excited to be starting my work on this project and getting involved with a number of really excellent collaborators and just learning more about trees. So with all of that, oh yeah, and so I was fortunate enough to go out this summer while I was wrapping up my PhD. I came for a little bit of sampling, and these are some of the pictures that I took this summer from the experiment. So these are lots of funky different Lepidoptera, and so this was really cool to see the experiment in action. And it's been making me think a lot about my undergrad work at William & Mary, where I was studying these butterflies, so a different life stage, but yeah, I just feel really grateful to be doing what I love for a little over 10 years now, which is kind of crazy too. So with all of that, I would like to acknowledge all of the folks that helped with this work, collaborators, funding sources, the excellent undergraduate students I worked with throughout the years, and I would be happy to take any questions. Thank you.
Yeah, that was a very nice talk.
Thank you.
Good job. And so one aspect of plant insect interactions is that nitrogen content is one of the most limiting elements for herbivores. And that's kind of the reason why legumes, for example, that have nitrogen fixing rhizobium nodules are more herbivorized than, say, those that don't have rhizobium nodules. And given your three experiments, could it possibly be that that below ground plants have lesser nitrogen content, absolute nitrogen content, than the above part of the plant, and that could be determining some of your results?
Yes, that's a great question. And so yes, definitely the results with the protein content in the leaves of the plants were quite surprising.
I noticed that.
Yeah, the protein's going up, which you think typically is a limiting nutrient for herbivores. So it was strange that they weren't excited about that and kind of going for those. So Yeah, still not entirely sure what's happening there. It's possible that they're just regulating more for carbs. There are species-specific instances of insects who prefer kind of a carb-based diet over protein. So you'd really have to get in there and do some experiments with different nutrient ratios to figure out what exactly they're targeting, which we didn't have time for in part of this work. But yeah, and then in terms of your thought about reallocating nutrients, so that has been documented in the literature before and is again thought of as this kind of tolerance mechanism of the plants where if you have a below ground herbivore, perhaps you move the stuff that maybe they're after above ground to maybe deter them or kind of get them to stop feeding on you. So yeah, it's possible that that's what we're detecting with that increase in protein, that the plants are shifting it away from the larvae essentially.
So it might be the stoichiometry of these elements that may possibly be important.
Yeah, yes. Exactly. Yeah. Thank you. Great question. Yeah.
Do you think there's any relationship between these results and the fact that the adult cucumber beetles are chewers and the bugs are piercing, sucking? Does that have any? Have you been able to tease that out at all?
Yeah, that is a really good question. Have we? I'm not sure we really tease that out so much in this particular collection of studies. But what I will say about the squash bugs is that they're, I also think of them as like kind of weird piercing sucking herbivores. So they're Koreans and they have giant stylets that they're just like jamming into the plant tissue. And they actually, which is way different than like an aphid, right? Yeah. So aphids have really tiny stylets that can oftentimes kind of move in between plant cells as they're kind of wiggling their way to the vasculature. So but the squash bugs is totally different. They're just kind of probing around and you can actually see some of the damage that they're inflicting on the host plant itself. So yeah, I'm not entirely sure if I mean, obviously they're using very different
I'm noticing like kind of a mostly liquid diet versus one that's actually
right,
able
exactly.
to consume part of the foliar material.
Yeah, exactly. So yeah, and that's the thing with the squash bugs is that we don't exactly know what they're feeding on. So it seems like they're probably hitting the phloem and the xylem and probably some plant material on their way in with their giant stylets. So yeah, it's but it's a good question and something I think to think about moving forward.
Following up on Matt's question, if you look at piercing and sucking insects versus chewers, piercers and suckers take out a lot more photosynthate than chewers. So even though the damage on the surface may be minimal by just looking at puncture marks versus maybe an excision on the side of the leaf for hole feeding or even surface feeding, the amount of damage done by piercing and sucking insect is woefully underestimated compared to chewing insects.
Yeah. Yeah, that's a great point because we kind of have a visual bias, I guess, to a certain extent. Yeah, and when the squash bugs aggregate, and particularly when the plants are smaller, they can really just knock them down, and they totally lose trigger pressures, wilts. So yeah, I think that's a really excellent point too. So yeah,
I don't think we've seen that.
Yeah,
I think it's the soil and how.
I think was just asked.
What do you think? I don't know.
So,
I don't know either.
Does it seem like potentially the reason that they might preferentially deter the squash bug is because they might do more damage? Is that okay? Yeah.
Yes.
But then also, is there like any sort of benefit provided to plants from the larvae feeding on their roots? Like, is there like a biodegradable benefit, or is it pretty much all negative?
Yeah. Excellent questions, Kat. Kat is not an entomologist, so this is very savvy for an entomology adjacent, I guess, person maybe. Yeah. Yeah. So, oh man, and I had yes. So yeah, your thought about your first question. Oh man, and I'm blanking out. But I had a thought about. I'll just start talking about what my thought was. So. The other element with these herbivores, as I mentioned, is that they're also vectors of plant pathogens, 2 different plant pathogens too. So that's like a whole kind of third element in this equation that we didn't quite get to in any of this work. But yeah, I also wonder if the plants sometimes are more worried about the pathogens that they could be carrying than the herbivores themselves too in some of these interactions. So something interesting to think about. And then in terms of could there be a benefit to plants when herbivores are feeding? Yes, there. So I didn't talk about it, but basically tolerance is kind of thought of as the plant being able to sustain growth after herbivory to a level comparable to a non-damaged plant. But there are instances in the literature of this phenomenon called overcompensation. So the plants actually grow more and do a little bit better, or they make more flowers or more fruits. I don't think that we ever really found evidence of overcompensation per se, but it is definitely something that's been documented in other systems. So it's a good question. Yeah, very cool. Yeah. I think there's a Zoom person maybe who, if you want to chime in, who's raising their hand.
Yeah, that's me. That's Bill Cooper. I used to be up there. Well, up there I was a butterfly photographer. Down here in Florida, I'm actually doing all sorts of other stuff. But this is a fascinating talk. I'm actually have a little NGO that I'm trying to get going off the ground in Costa Rica to do some reforestation. So I started on a paper on tropical forests. It's now up to 315 pages and about 800 references. But one of the interesting areas that I got involved in writing this is herbivory and how complex it is. But the one thing that was missing in the soil are the fungi. They're so critical to the growth of plants. I wonder how they fit into your story.
Yeah, that is a great question. Yeah, thank you for that question. And so one of the folks in the PhD lab that I was in that I just left, he is working on a beneficial fungus that associates with plants called arbuscular mycorrhizal fungi. And this particular fungus has oftentimes been shown to enhance plant defense against herbivory. So yeah, I think there's going to be hopefully some work coming out of Angel's lab from Texas A&M looking at some of those beneficial fungi in the cucurbit system. And soil microbes have definitely been on my mind. I had another project that I didn't talk about today where I was looking at fungal communities associating with plants as well as bacterial communities in the soil and how they're both enhancing plant resistance against herbivory, but then also how insect feeding damage is altering the community that's associating with plants as well. So you kind of get these two different mechanisms that the microbes could be playing a role in plant-insect interactions, either kind of being the drivers or the ones responding to plants that are damaged by herbivores. So definitely really interesting things to think about moving forward. Yeah.
So actually, those arbuscular mycorrhizal fungi are very interesting because they also produce a chemical known as glomalin, which we really don't know what the structure of that is, but it's actually very important in carbon sequestration because it actually can have a half-life in the orders of 40 years in the soil. Somebody also asked me where I'm from in Florida. I'm at the University of Florida right now in the Department of Environmental Engineering Sciences. But Morgan, I'd like to get in touch with you. I see your email here because I just like to talk to you about what you're doing. I think what you're doing is fascinating, and I think you're doing a great job. And I love the enthusiasm.
Thank you. That's very sweet. Yeah, feel free to email me anytime. Thank you.
Okay.
Yeah.
Was a minor plot point, but I'm curious, why did you use mated pairs for the squash beetles in one of your experiments, but not for the other beetles?
Yeah, that is a really good question. In thinking about which herbivores we were using for the above ground plant preference assays, so we were focused just on female beetles because the male beetles actually emit an aggregation pheromone. And we had some other work in the lab that Don, I think he's on here on the talk. We had a collaborative project with Don that I think he touched on at ESW meeting a couple weeks ago where we were looking at plant responses to exposure to that aggregation pheromone. So we didn't want that variable interfering with these particular experiments. So we focused on the females, and the females eat more. So we just wanted them to chomp on those leaves. And then for the squash bugs, we introduced mating pairs to look at oviposition, essentially. So we wanted to make sure that the females were mated, and if they weren't mated when they were introduced to the arena, they were gonna mate and then hopefully lay eggs in the experiment. So yeah, but good question. Yeah. Yeah.
So it was working and living with cucurbits for years, like changed their own herbivory. Loving more squash recipes, or you never want to see another one.
Yeah, that's a great question. Yeah, I do like the plants that I work with, and I had a field experiment with the watermelons one summer. That was great. We had all these watermelons that we were giving out to folks. And yeah, and my philosophy is you should pick a study organism that you really want to eat, because it's nice. But once I had a field experiment that I didn't present on, but with the zucchini plants, and man, once they start producing fruits, they just are going nonstop. So there was one summer that I had hundreds of zucchinis that I was trying to give away to folks in the department. So that was a bit of zucchini overload. But after that summer, it was good. So yeah, that's a good question.
We had one question in the chat.
Oh, sure.
From Robin. I can read it out. Robin asks, for the HIPV plant communication experiment, do you think there would be a difference if there were some bins where the larva damaged emitter plants?
Sorry. I can pick up the rest of the question. Where we left the larvae damaged emitters in the bin. Yeah, thinking about how this could be translated to an applied system. Yeah, that's a great question. So the rationale behind, so typically when you're conducting these plant communication experiments, the rationale behind removing the plants is to definitively know that it was something about that prior exposure that's changing the response in the receiver, rather than something that your emitter plant is doing. If it's left in the bin, it kind of becomes a confounding factor, right? Like, is it the response in the receiver or just the emitter still being there that is influencing whatever's happening in your neighboring plant? So to control for that, that's why we removed the emitters from the bins. I think it would be interesting definitely to think about possibly what happens if we leave the emitters in the bins. And then in terms of application in a field, something again that I didn't quite get to in my dissertation, but I think it would be really interesting to just test osamine dispensers in a field, right? So if we put out some osamine dispensers, do we see an enhanced plant resistance against squash bugs? Could that even translate into some boost in the yield of the plants? So as a chemical ecologist, I say that's always my dream, is to find some naturally produced chemical that can make a farmer's life a little bit easier. So I think definitely kind of keeping the applications in mind, I think is always key. So yeah, thanks for the question. Yeah, yes.
This is very dumb, but where do you get like osamine online?
Yeah, I don't know exactly where we ordered it from, but just some chemical company. Yeah, you can order some of these compounds, and then we just took a little bit of it and diluted it in dichloromethane, which is very evaporative. So and measured the rates too to confirm that it was emitting things at the rates that we wanted. So you can order your own volatiles online. They have a bunch. Good question.
Was that question where you found the mycene? Is that what the osamine? I mean, was that the question?
The question was, where do you get osamine?
Yeah, so I'm sure that would not be a hard question to answer. You just go into Google and you put that in. It's probably something like a Sapelco or which is up by Penn State or another place. I'm sure it's interesting because it's just an isoprene, two isoprenes hooked together.
Right. Exactly, yeah.
Did
Yeah.
you determine that by GC mass spec then, or just GC?
Yes, I was working at, sorry, it's a little hard to hear folks in the room. So I think I heard your question and I was working with a GCMS.
Okay. Yeah, that's the answer.
Yeah, okay. Any other questions?
Thank you very much.
Thanks, everybody.
All right, if there's nothing else pending, I would like someone to make a motion to conclude the 1,251st meeting. Alita, seconded?
Second.
So moved. Thank you, everyone. There's still some pizza left. There's some soda. Please pay if you can. And if this didn't make you hungry for food, the next month's meeting will be about ants and fungus farming, I'm sure. So that'll make you hungry too. So stay in the zone.
Fungus farming.